Kick it off. All right, we will be recording everyone. Hope you're okay with that. Please feel free to join. We'd love to see your face, but if not, being a mute and off camera is fine as well. We are excited today. We have a number of fun and exciting things to be sharing with the community today. Our two big topics are gene editing. There was a big win in the field a few weeks ago that we're all still celebrating. We wanted to share that win, kind of what happened and what it means for us as a community. And then we have an internal win that's very specific to SCN2A. It was recently announced that SCN2A will be awarded an ICD-10 code. So we'll be explaining what an ICD-10 code is and why it's important, and then what you can do to help going forward with that initiative. Huh. So I guess what we'll be going through, guess I kind of mostly touched on all this, but gene editing of baby KJ, what happened, what they developed to help, how long did it take, and why it's important to the field, and what it means for us as an SCN2A community. And then for ICD-10 codes, what is an ICD-10 code, why it's important, why it was such a battle for us to get. It took a long time, and there were a lot of people putting in a lot of work to get this done. So thank you to all of those. And then what our role is in it as a community to start using this ICD-10 code. So baby KJ. Our recent news that came out is there was the very first ever personalized gene editing that was deployed to a patient. And it was a fairly newborn little boy named baby KJ, and he had a rare condition. You could see him right here, and he's doing well in this picture, but we'll kind of take you through his journey. But you can see all the smiles on the family's face celebrating this major win. And I can imagine any of us kind of getting a big win like this would be absolutely through the moon. But this was baby KJ when he was born. And what happened with him is he was born and almost immediately he was experiencing extreme tiredness or lethargy and was having a lot of respiratory distress. And when they looked in his blood, they noticed that his ammonia levels were really high. And that's a big warning sign because when your ammonia levels can be pretty toxic when they get pretty high in your blood. So that was a warning sign for the clinicians to start doing some additional testing. When they did additional testing, what they found is that baby KJ had something called CPS1 deficiency or carbamoyl phosphate synthetase 1 deficiency. And this enzyme, what it does is normally we all make ammonia from eating food and digesting it and with different metabolism processes that we go through. But that ammonia isn't allowed to accumulate and become toxic because it is enzymatically broken down and converted into urea, which is your pee. And then you excrete it out so it doesn't become toxic. And in instances where we have, or an individual has mutations in this gene, they're not able to metabolize ammonia into urea, and it accumulates in the blood and it becomes toxic. Um, his initial symptoms of lethargy and respiratory distress are kind of the initial symptoms, but it quickly turns into seizures, coma, and death if it's not treated. So it's a very serious and life-threatening condition. In this particular instance, it was caused by two mutations. So the majority of our families and our kiddos are due to just a single mutation, one bad stroke of bad luck. Baby KJ actually had two. And it's because one single copy is sufficient to metabolize and not be toxic and be lethal. And it's only when you get both mutations that it becomes lethal and toxic. For us with SCN2A, SCN2A is so important that even a single mutation causes the symptoms that we see in our children. But that's not the case with this indication. But again, we'll kind of get into why this type of mutation is important because we see it in our community too, these truncating mutations. So we talk about our P's and our P dots and C dots, but a lot of us that have loss of function are due to this X, and that means that X on one of the alleles passed down from your mother or father isn't being produced into a protein. So from baby KJ's instance, he's not getting protein from either his mother or his father. So he essentially has none of the carbamoyl phosphate synthetase 1 enzyme. But what's really interesting and exciting about this whole process with him, we're celebrating that, it was a successful gene editing. But what's really exciting is how quickly they were able to do it. So baby KJ had symptoms. They were able to get a genetic diagnosis quickly. We've come a long way in that. There's a lot of people in our community that had a long diagnostic odyssey, but I think we're doing quite a bit better in that regard of late. Um, but they were very quickly able to get the diagnosis, develop a patient-specific cell line, screen for different drugs, and kind of essentially kind of engineer a very specific drug for this particular patient. So in a very N of one centric way. They quickly turned it around into a ad hoc mouse model with a very short turnaround. And then they began testing this. They got in front of the FDA, which is our United States regulator that makes the determination of whether a drug can be tested in humans or not. They got a really quick turnaround and approval from the FDA saying it was okay to test and to go forward. They then tested whether their drug that they made was safe in non-human primates, and it looked like it was safe not only to be dosed once but to be dosed multiple times. And that study in the non-human primates gave them confidence in what dose they should be treating baby KJ with. Um, and then they prepped the drug, got ready to go. Submitted it for final approval by the FDA and got that. And then they dosed baby KJ once. It wasn't enough to get him totally to where they wanted to be, and then we're able to dose him a second time. And that second dose is an important element that we'll get into in a moment. But I do want to highlight it here just to kind of, flag that for another part of our conversation, because how we deliver these gene editing and gene therapies, there are some modalities where we can only deliver once. It's a one and done. And there are some where we have the flexibility of being able to dose multiple times. And that's an important consideration as we think through how we develop drugs for our community and also which clinical trials we consider for our child. So for this particular indication, CPS1 deficiency, kind of our options that are on the table when we think about how we can help baby KJ or individuals like him. So again, it's caused by this deficiency in this enzyme and this accumulation of ammonia in your blood. So the main objective is to lower that ammonia in the blood and do it as quick as you can. And there are some medications that can do a decent job of that and stabilize it to some degree, but they're far from curative. And those are called ammonia reducing medications. And baby KJ was quickly put on that medication, which is glycerol phenylbutyrate. And you also want to modify your diet to decrease the spigot at the other end that produces ammonia. You want to make that as low as possible while still having enough nutrition to be able to thrive. So they also changed the diet for baby KJ as well. This enzyme isn't a very straightforward, or there are multiple processes in the conversion of ammonia to urea. And one of the steps downstream of CPS1 is citrulline. So you can also supplement with that, which can also help reduce ammonia and work through that process. And they tried all three of these with baby KJ, and it still wasn't sufficient to get him to goal, which is the case for just about all individuals with this severe CPS1. So the next step that is only really available to these individuals is a liver transplant. And a liver transplant can be incredibly helpful. And the reason why that is because you're bringing someone else's liver cells into your body. And those liver cells do have CPS1, and they're able to metabolize that ammonia and clear it out of your body. So it can be very helpful. But with a liver transplant, you're taking somebody else's liver and putting it in your body. And that requires immunosuppressive drugs. And there are a lot of side effects and consequences of having a transplant like that. And it's not a guarantee that it'll take with your body. So it's still a very high risk procedure, even though it can be very beneficial. So when we think about kind of risks and rewards, the risk of not having a liver transplant, it definitely is much higher than taking a liver transplant. So most individuals elect to go that route. But certainly looking for better options. And that's why, gene therapy and genetic medicines make a lot of sense in this scenario. For genetic medicines, we're entering a really fun era where there's not just one gene therapy. And we hear about gene therapies a lot in the news. And I think sometimes we think that there's just kind of one approach. That's not the case. There are now a whole bunch of tools in our toolbox of ways that we can either replace a gene, we can edit a gene, we can tinker with a gene, and depending on what is happening in that particular use case, we want to pick the right tool for the job, so to say. You don't want to be pounding in a hammer with a screwdriver, and you don't want to be putting in a screw with a hammer. So depending on the situation and the use case, different genetic medicines are appropriate. But for this particular instance, they elected to go with gene editing, which is a more novel approach that's been around just for a handful of years, and only a few patients have been treated in the clinic with this modality. But for CPS1 at prior to baby KJ, it was, totally theoretical. So how does gene editing work? Well, it uses a few tools that are working intimately together in order to complete the job. These tools, and I think as we as a community start to really rev up with gene editing and, some of these come online, which will be really exciting. Some of these terms will be important for us to just kind of remember as we begin to talk about these more and more. Um, but the important kind of components of this tool, the first is called a guide RNA. And this is kind of like a targeting piece of this medicine. And it allows the medicine to be specifically targeted to the mutation in your DNA in order for it to be able to correct it. The other two important elements are one is called a Cas protein, and in most instances, it's a protein called Cas9. But how this works is it's tethered to a base editor, and these two work in parallel to edit the DNA where there's a mutation that's pathogenic. So the Cas9 protein goes in and cuts just one strand of your double-stranded DNA, and that cut in the DNA releases some signals that it has to be repaired. Breaks in the DNA are a big kind of warning signal for our system and our cells, and there are enzymes that quickly go in there and try to repair that nick in the DNA. And having the Cas9 protein that cuts it tethered to a base editor as one of those correcting genes allows it to be a really efficient system. So the guide RNA brings these specifically to the mutation. The Cas protein then cuts the DNA, which alerts the body that it has to be, fixed. That break in the DNA has to be fixed, and then the base editor flops out specific base pairs in the DNA for other base pairs. So the idea is that you want to switch out the mutant base pair for a different amino acid that isn't pathogenic. So that's roughly how these gene editors work. And we as a science ecosystem are getting quite good at this. We're getting really good at using these genetic editing tools in a lot of different cells. The problem really is all on delivery for this point. So we have the tool, but then we have to get it to the right organ system, and we have to get it to the right cells. For the instance of this particular indication, the liver is the target organ. And scientists, the liver is kind of the furthest ahead in regards to genetic medicines. We've gotten really good at delivering different drugs and different genetic medicines to the liver. We understand what the target is that allows it to get to that organ specifically. So ways that different delivery vehicles that are available to us that are the most commonly used. One is a virus. So you can package this tool up in a virus, and the virus goes into that target organ and infects all the cells that you want that tool to be used in. But then there's also an approach called a nanoparticle, and usually these are lipids. So kind of fat, a whole bunch of fat that aggregates together, and on the inside of that you have this tool. And then you give this delivery, and it goes to the target cells, and it gets absorbed to the appropriate cells. So tool, delivery vehicle, and then target organ. And that's what they did for this particular instance with this particular child. So they delivered it via nanoparticle to the liver. And what happened? So at 3 months, ammonia, which was the danger thing that they were looking at in the blood, really elevated at certain times. So at 3 months until a dose of 1, there are these instances where ammonia really spiked up despite being on all those medications that we talked about earlier that can still reduce the ammonia. But then after the treatment with the genetic medicine, it lowered that substantially. But there were still a few instances where it got a little bit above, and there was some other marker that suggested that they weren't quite where they wanted to be. And then they treated a second time with these nanoparticles to get to an even better spot. And one thing to mention is after the second dose, they were able to reduce some of the other medications that they were on. And we think about that a lot for individuals in our community when we try a new medication that's working. Like, can we pull off something else? And that was the case for baby KJ at this point. Is after 2 doses of this genetic editing, they were able to pull off some of those scavenger medications that reduce ammonia. So you can see him right here. He's looking pretty smiley, pretty happy baby, which is really great. But a really cool story and some really cool learnings for us. And I think the biggest two learnings for us as a community, this is the first kind of bespoke gene editing that was successfully delivered. So it's a big win for the science and drug development community. But the speed of development, I think, is something that's really remarkable. Within an 8-month period, they were able to, diagnose this patient, identify a drug, create that drug custom for this child, test it in not only cell lines, but mice that were specific to that patient, work with the FDA, treat non-human primates, and then deliver multiple doses, but all within that time frame. That's just absolutely remarkable how these timelines that used to take years and years and years are collapsing. It's because we're getting really good at knowing how to use these tools and build these models that we can test these different drugs in. So two really uh, big important wins for the drug development and drug development community. But what does it mean for us? Why is this important for us? It's hope. It's confirming that we're getting really good with these genetic tools, and particularly with gene editing and different gene therapies as a science ecosystem. But the liver delivery is ahead. It's more advanced than it is to the brain. But we're getting really good at brain too. We're hopefully going to be a fast follower with some of these discoveries that we've made with the liver and how we've been able to deploy them. A few of us on this call from our Tasco team were just at a conference looking at how we could deliver to the brain more successfully, and the data looks really promising. And these are going to be going into humans and a few other indications in the very near term. And if those prove to be successful, it'll be a really big watershed moment for CNS and genetic therapies as a community. And it could really speed things up and bring a lot of attention to CNS gene therapies for us going forward, which is great. So if that happens, that gives us a new tool to be able to deliver to the brain. And we could put all these different therapies that we've been working on, and it'll kind of rev up a lot of different drugs that we have in development for our community. And just wanted to refresh our community of all the different things that we have available, and things in development for our children. For seizures, there are 9 drugs in the clinic right now that are enrolling. So these are new novel mechanisms of action that could potentially be beneficial if you tried a lot of things and still haven't gotten control yet. I know with our daughter Harper, we still have daily seizures despite being on like 5 different anti-seizure meds. So considering different modalities to get better control are always really important. So a lot of drugs in development for seizures. There's a handful in clinical development right now for autism and behaviors associated with autism that you could enroll into. Shayna, are any of those specific to SCN2A related seizure types? None of those are, but these are. So the first two slides here are more kind of symptomatic type of drugs that are enrolling. The next few slides will all be SCN2A specific trials. And we often bucket our children into a few different clusters, gain of function, loss of function, and mixed function. And we think about our therapies in that way too. For gain of function, kind of our approaches are either precision next generation sodium channel blockers or knockdown and knockdown replace strategies are all make sense as a means of, if it's gain of function, we want to bring things down. There's drugs in development by Praxis and Neurocrine that are in the clinic right now for sodium channel blockers. So these are oral small molecule drugs. And then for kind of precision genetic medicines, there's a drug in development by Praxis. There's a drug that's in the clinic that was developed by Nlorum, and it's being deployed through Rush University. And there's also a promising drug that could potentially affect all of our community in development by University of Iowa or UNC for gain of function. For our mixed function, there's a different drug in development or different drug in the clinic that was developed by Nlorum. And this is an allele-specific antisense oligo for mixed function. And there's that same drug by Sam Young out of University of Iowa that could potentially benefit our whole community. And there's a few drugs that are testing compensatory mechanisms that could potentially be beneficial for our mixed function community. And for loss of function, there's more tools in our toolbox. So these are people that have a haploinsufficiency or they have that X similar to baby KJ. There's upregulation technology makes sense. Replacement and knockdown makes sense. Gene editing and correction makes sense. And we have one in development right now, and there are a few other strategies. All of the loss of function are currently in preclinical and discovery right now, so none of these are in the clinic. But there are a lot of shots on goal that are actively in development right now, which is really exciting. But if that delivery modality is validated in these next few clinical trials, it will be very enabling for a lot of these that are still in proof of concept. Because once you get proof of concept thinking about the tool, if we have a delivery approach that works, then we can package them together and get them into the clinic. Yeah. So I will pause there on kind of baby KJ and kind of the exciting news there. Happy to take any questions on that real quick. And if not, we'll move on to Melody and talk about an ICD-10 code that we have.
Thanks, Sean. Feel free to take yourself off mute and ask questions or put them in the chat, either way.
And I can move the slides for you, Melody, too.
Wonderful.
There's
Thank
one in
you.
the chat, Sean, for you if you want me to read it quickly from Shayna Wood.
Sure.
I think you
asked,
already answered that one.
Oh, did you?
Yeah.
Oh.
But just to answer it again, those last few slides were SCN2A specific, Shayna, and the first few were symptomatic treatments that were in development. All right, Melody. Ooh, yeah, absolutely. I think Leah's recording this, and we'll have it on our website afterwards, and we'll share the slides as well.
I have a question.
Go ahead.
This is really definitely great news. Thank you for sharing. I was wondering, for baby KJ, they use the CRISPR technology, but that isn't, my understanding is that isn't appropriate for the SCN2A. So are they working? And the conference I attended in Boston, I spoke to, I forget his name, the researcher at San Francisco. He said, he had developed a treatment for loss of function, but we didn't have a delivery mechanism. So what is the work being done for delivery?
Sure. And I will
Alternative modalities besides CRISPR.
Yeah, absolutely. So CRISPR is the tool, and the tool then is delivered with either viruses or nanoparticles. And I wouldn't say that CRISPR isn't appropriate for 2A. I would argue quite the opposite, that CRISPR makes a lot of sense for SCN2A because we have so many variants, and we have in this trichotomy of mixed function, gain of function, and loss of function. But the work that's being done to help deliver our tools, which could be CRISPR or the gene. The problem is something called the blood-brain barrier, which prevents drugs from getting to the brain. And there's been work that's looked at a transferrin receptor that if you target that transferrin receptor on either your virus or your nanoparticle, it facilitates the passageway through the blood-brain barrier. And there's been, quite a bit of work done on that particular receptor, and it looks really promising. It's work done by multiple parties independently that have all shown that it can enable really widespread delivery to the brain. But they have to prove it in humans. We can show it in mice, and oftentimes things in mice look good, and they may not look as good in humans. But I think a lot of the data looks quite promising. Karen, do you have any thoughts on that modality and whether it looks good or not in your opinion?
From the data that we saw in New York at the conference that we just attended last week, I would say that there are many promising avenues that independent laboratories are working on simultaneously to increase the access that CRISPR would have to the brain. And I think that's a very hopeful step. We can't hit every single cell in the brain at the same exact expression level right now. And I think that is the challenge that these investigators are trying to address.
Okay, that makes sense. What is the timeline? Do you guys know?
For proof of concept for this new delivery approach, we'll probably find out by end of year whether it's successful or not. and then how quickly those could start to be translated and adopted into the drugs we have in development. I imagine if it's a big win, everyone will. It's often kind of a herd mentality with drug developers, so if it's a big win, everyone will try to adopt that approach, and it will bring a lot of excitement and money to, those development programs, which is very enabling.
Mm, that is great. So another quick follow-up, not specifically to this, but given everything that's happening at the NIH and all that and the cuts to funding, does that impact the work on the SCN2A gene? Or I have no idea if it's private folks or if there's any NIH funding or how it would impact our community.
Karen, do you want to take that one?
So in my private conversations that I've had with various scientists who do focus their laboratories on SCN2A, I would say that everyone across the board has been affected. And as a drug developer who worked in a pharmaceutical company myself, that we received an NIH grant that is in a critical position right now of possibly being cut. Universities across the board are suffering from the cuts in indirect funding from any NIH grant that they have been granted. For the most part, DEI grants or any grants that have contained words that might be interpreted as involving DEI have been cut first. So that doesn't directly affect SCN2A, but I think there is overall deep concern starting from the heads and ex-heads of the NIH and the ex-head of the FDA all the way through the community and even through the therapeutic industry. So there's across the board concern. And I would say that as a foundation and as members of a concerned and active community, we should be keeping tabs on this and supporting our researchers in any way that we can.
I do think that, grassroots fundraising and us being able to support these programs through these elements are really important. So I think this is like an opportunity for us to, really kind of hit the ground and try to raise funds internally to continue to support these. And I think one trickle down is, there are a lot of really bright, brilliant researchers out there. And when everyone kind of loses a little bit of money, it makes that, better researchers. We have a bigger pool of researchers that we can grab and fund. So I think there are opportunities where we can get in front of like the best and the brightest researchers out there if we have money to dangle. So I think it's important and incumbent upon us as a community to fundraise and continue to really support research.
Thank you.
Thanks
Any
for
other
raising it, Krish. I think it's an important topic. If there aren't any more questions on baby KJ or the gene therapy, maybe we should move on to the ICD-10.
Yeah.
I had one question, Leah, if it's okay. I was curious, long time ago, I want to say like 5 years ago, I listened to like an SCN2A, one of our podcasts. I don't know, it was something a while ago. And they were saying that for SCN2A at the time, the thought was that it would be easier to do to manipulate like a gain of function gene because it's easier to take something away than to create something that isn't there, is like what I got from it. But it sounds like this is like something that's focusing on a loss of function. So I was just curious, like what is it about the loss of function that makes it like attractive for this gene therapy? Because I always just kind of, and it made sense to me with the gain of function. I have a loss of function child, but I just really curious about why like they're now thinking that they could help the loss of function community.
Yeah, absolutely. I think a big part of it is because new tools have been developed. This is a good example. But as we think about different modalities, and I can even go back. So for loss of function, we have the opportunities to upregulate or turn on. For SCN2A, it's often a de novo. So you have your gene given or your allele given by your mother and your father. Usually it's just one broken for 2A. So we have the opportunity to upregulate from the healthy copy. So that puts us in a unique position. we can replace and knock down and replace for loss of function. So you can take the broken gene and replace it in some way, shape, or form by delivering a new gene. So that opens up a class of drugs that can deliver the gene. There's some nuance on that because 2A is a big gene, but we are working on different strategies to deliver big genes. And then with the CRISPRs, we can now edit and correct the genes. And there's one. This is one approach, but there are multiple versions of gene editing that are coming online. So it's a class. There are multiple kind of buckets underneath there. But I think another important element that's bringing a lot of people to the table is it's now well regarded that SCN2A loss of function or SCN2A gene at large is like the leading monogenic cause of autism. So as that research continues to mature, being like the leading genetic driver of autism has brought a lot of awareness and excitement to be able to correct SCN2A.
Oh, you signed me up for that trial.
Thank you for the question, Victoria. All right, I see Melody chomping at the bit down there.
Wonderful. Can everybody hear me okay? Can you guys hear me?
Yes, we can.
You sound
You can
great.
hear me? Okay, fantastic. Thanks. I apologize. I am sitting in the Philadelphia train station on the floor in a corner looking for a quiet spot to say hello to everybody. So apologies for the sound. It's actually really exciting to be. I'm 5 minutes away from where baby KJ was just dosed, and it's a real there's a buzz. There's a buzz in the community. It's really exciting. I'm actually up here for a conference from another foundation and learning their ways of engaging with the FDA. They just had a PFDD meeting that I attended today, which was fantastic, and bringing that back to the community. But today we've got some other exciting news. I'm here to talk to you about the new ICD-10 code or ICD-10 CM code, which is really exciting. So if we could go to the next slide, please. Okay, so this is a really big announcement. Effective October 25th, I'm sorry, October 1st, 2025, SCN2A related neurodevelopmental disorders will have their own unique code. And this is really special and very rare in the rare community. And it's all thanks to incredible advocacy efforts from the Families SCN2A Foundation long before I came to join TASCO. So I will pass it over at some point to have Sean or others in the foundation explain all of the work that went into it before I came in, but I'm here to talk more about what it means to the community and how you can help. So next slide, please. Fantastic. So just a quick, there we go. So what is an ICD-10 code or an ICD-10 CM code? So when you go get medical care, your healthcare providers are going to be using uniform codes to describe the conditions or symptoms that you have. And that helps to keep your records clean. And so they can understand, different providers can understand what conditions you've had or your child has had. But it also helps for medical research and public health surveillance and lots of other stuff that we'll talk about. These codes are used all over the world. The ICD-10 itself comes from the World Health Organization, but ICD-10-CM is actually a United States variation of that through CDC. So you can go to the next slide. I think we talked a little bit about that as well. So as I was mentioning, ICD-10-CM, tongue twister, in the United States is used in the United States. There are some other countries, I believe, that adopted as well, but generally it's a US modification. There is already an ICD-11 out in the world, but many countries have not adopted it. And as far as we know, the United States is going to be using ICD-10-CM for the foreseeable future. Next slide, please. So why do we need our own code? So as I mentioned, there's thousands and thousands and thousands of ICD-10 codes, and some of them are extremely specific, as I'll give an example in a moment. But many rare disorders like SCN2A don't have their own code, and they just get lumped into generic categories. And so some medical facilities may code it one way, and some may code it another way, and there's no uniformity. So it's really hard to kind of track this health data and to figure out what's going on in a community and try to find folks. For example, I was even looking up just to see what would happen if I typed into to Google search and I said, what is the ICD-10 code for SCN2A? And every time I typed it, I got a different answer. Which depends on what AI thought of at the moment. So it's really exciting that there will be a set code that all coders in all medical facilities in the United States and hopefully beyond will start using to try to make sure that everybody is captured. Next slide, please. So just to give you an example, this is actually a screenshot of an actual SCN2A patient. And this is how their provider coded them. So they had an abnormal wellness exam. This person had scoliosis. This person has seizures. This person has a genetic defect, nice random generic term, and some gastritis, and uses a wheelchair as an ambulatory aid. And so this is, it tells you a decent amount about this person, but it's very non-specific. This could be SCN2A. It could be dozens, hundreds, thousands of other conditions. You wouldn't know just by looking at this what this person is experiencing. And on the next slide, I'm going to give you an example of an extremely specific one. This one's going to go through a couple pictures. So first picture, that's me. Hi, this is me at the petting zoo with my children, and it was a gift from my daughter to go see the capybaras that she loves. And we went there and we met a pig that said, please feed me. So I did. But apparently the pig didn't want its kibble. The pig wanted my blood, and I got a significant bite on my finger. And as you can see, I have an extremely specific code. So anybody who was doing research to find out, how often do pigs bite people on their right middle finger? There's a code for it, and they could search anonymous data and find out how often do pigs prefer right hands to left hands? Do pigs prefer middle fingers to thumbs? I don't know, but it's in the code. So this is an extremely specific code that is just not, it's hilarious. And I saw the comment, and not only is there a code that's specific for being bitten by a pig, but it remains in my medical record years after this happened. I don't know if they're keeping it in there in case, 5 years from now, I have some random disorder and the infectious disease doctors are like, oh, she did get bit by a pig 5 years ago. But every time I see my medical records, I laugh because it's pretty funny that it's now part of my record. So extremely specific, perhaps more specific than they will ever need for research or for medical care, but that's an example of a highly detailed one. And I showed you an example of up until now, how not detailed an SCN2A code would be. So you can go to the next slide, please. And so I showed you that in the beginning, we've got these numbers. So it's the QA 0.0101, and I just want to show you it comes from different classifications. And so if we can kind of click through the slide, it'll show you the first, like, what does QA mean? And then what does QAO mean? And then what does QAO1 mean? And 0101, and it shows you more and more and more specific. So not only do you have the same code for every person who's using it, but anybody who knows that code can actually work backwards and see under which general umbrella this falls. This is a channelopathy, this is a neurological disorder, this is a rare disease. And so you can learn a lot just by looking at the codes and going backwards. Next slide, please. Okay, so using the new code, one thing that's really important is when your provider starts using this new code, it's not meant to replace anything else that's already there. So codes for autism, codes for seizures, codes for any other conditions that are going on, those still stay. You don't just code one, but it's giving an additional layer of information. And so the reason why it's super important not to have it replace anything is that it's a new code. And so one, you'd lose some detail if you just had that. You wouldn't know what kind of SCN2A related disorder if you just use that code. But also your insurance company or your payer may not know what that code means, and they may not have already connected it to payment. So for example, if your child is getting therapy, getting physical therapy for a symptom related to their condition, and the code is for something that the insurance companies know, oh, if this child has this, then physical therapy is an appropriate treatment, then it may be on automatic that they'll pay for it. Whereas if you started switching codes, they may not realize and it may delay. So it's really important to have it as an additional code, not as a replacement code. And so we can go to the next slide. Please. Thank you. So here's an example of new coding. I made this up just based on, chatting with some folks. So if somebody had an SCN2A related epilepsy, their codes they may have multiple codes, as I showed you in the other one. So the first one would be SCN2A related neurodevelopmental disorder. That's great. But also coding for the seizures, also coding for wheelchair use. And then you may see some random codes in there that may or may not have anything to do with SCN2A related disorders. May have, this one's somebody who's, gets a runny nose from cat hair, right? You may have other conditions and they may not be related, but maybe they are too. And maybe we won't know until we're able to start really looking at lots of people with SCN2A related disorders and their other conditions and saying like, hey, it seems like there's a cluster of kids who are taking medication or kids who are showing this other comorbidity or other signs or symptoms. And it's seemingly unrelated, but maybe they are actually related in some cases. So it'll be really interesting to see. We can go to the next one as another example. Next slide, please. This is a hypothetical for somebody with an SCN2A related autism diagnosis. So again, it would start off with the cause, the new code, and then a code for autism. And then maybe this child also has scoliosis, which often goes along with other conditions. And maybe they also have asthma, and maybe that's related, maybe it's not. They still be coded. So you see lots of different codes, but you code all of the major things together. So it's really exciting to be able to do this, and then I'll tell you, some of the things we can do with that. We go to the next slide, please. Oh, actually, I'm going to pause for a second and let somebody else talk for a moment about the very long process that it took to get this new code. Long before I came to work with the foundation, there were a lot of advocacy work going on. So if somebody else would like to jump in, or if we want to skip the slide and have that as part of the chat later, either way.
Leah, do you want to talk on it? You kicked it off.
Sure. Well, we knew that this was something that would be helpful for our community, so we put in an application in 2021. Oh my gosh, it's hard to believe it's been that long. But we were denied several times and just continued to keep it going. And then when Sean and Tasco took it over, they really put the oomph behind it, and I think also CDC was ready for making some changes finally. So got it across the finish line.
Amazing advocacy work. And I have to say this, on the tier of what rare advocacy groups do, this is a top tier win. This is not an easy ask. This is not something that many rare disease advocacy groups can pull off. So it's incredibly impressive that this team was able to put forth the application in such a way that it got approved and in a relatively short period of time by ICD-10 standard time. So we can pop over to the next slide, please. Okay, so how are these codes used? I had mentioned billing, and so that's a very common thing. The codes are used for your insurance company or Medicare or Medicaid or TRICARE or whoever's paying your medical bills. It's part of the healthcare billing process, but it's used for many other things, including research studies. And I put the little star there after talking to Jenny. I really appreciate her highlighting to point out that in the case of research studies and in many other uses of ICD-10 codes, your data is anonymous. They're not giving your medical records to people without your consent. It's anonymized data or de-identified data. For healthcare billing, it is identified, of course, because it's part of your payer. And for some research studies, it's identified, but you've given consent. You're a research participant and you're choosing to participate, but for others, I just want to let you know that people aren't taking your records without your consent. It also potentially can be used for clinical care. I'll talk about that in a second, and for public health surveillance. And then huge win. It really helps with the drug development process and also for regulatory review with FDA or the equivalent regulatory bodies wherever you are internationally. And then of course for advocacy work. So we can give some examples on the next couple of slides. So for healthcare payments, again, that was sort of the big use that most people think about when they think about ICD-10 codes. These codes help insurers or payers know what to pay for. But it can also help them as a group when they are doing analysis to understand the true cost of long-term care for these disorders. So if they're looking at SCN2A related disorders, and they can see years upon years upon years of surgeries or hospitalizations or potentially surgeries, I suppose, or rehab facilities or services, or all the different costs and cares and medications and specialists, and you start adding up those costs. When you look at it sort of on a long-term basis, it can help them realize that the cost of treating it symptomatically may be comparable to the cost of a disease-modifying treatment that they may otherwise not have been interested in paying for because of a high price tag. With new drugs, they tend to be exorbitantly expensive. But if you can show long-term the cost of the care untreated, oftentimes that can persuade payers to consider higher price treatments. It can also just generally increase the likelihood of coverage for a variety of conditions once they're able to start collecting data specifically to your disorder, and they get a better sense of what those costs may be. So we can go to the next slide. Researchers also can learn lots more about how SCN2A presents, but also some of those early signs and symptoms for people who later on develop SCN2A related or get the diagnosis. They've later on in their life develop the symptoms of the disorder. And so that can be really helpful to go backwards, and that could potentially lead to better screening or identify at-risk people and get earlier genetic testing, which could then improve outcomes by reducing those delays in treatment, or making sure that they're getting the right treatment if they understand if it's a gain of function or loss of function or mixed function variant. Next slide, please. And that rolls right into clinical care. That's care with your doctor or other healthcare providers. They can help track using these large data sets. They can help track which treatments and services and conditions are more likely to occur. Are people using the emergency room a lot because they're not getting good management in their regular visits? Are they getting hospitalized for infections that don't normally cause hospitalizations? Are they needing higher doses or more anything? What is happening in the clinical care? When you have a code that unites the whole community, the researchers and the clinicians can start getting a better idea of what the big picture is among all people with the disorder, not just the handful of patients that they see. So this can really help with planning and prevention. Next slide, please. And then so for public health organizations and agencies, government agencies will often use, again, de-identified data to help get a surveillance sense of the community. What are the incidence and prevalence and other statistics related to a disease burden, if you will. This data can also help everybody be counted, including people who either don't want to or unable to or are unaware of the ability to participate in registries or research projects or clinical trials or other data collection efforts. And so it can just be really fantastic way to capture a better sense of the whole community. Next slide, please. Drug development. So drug developers, the pharmaceutical, biopharmaceutical companies and the drug regulators like FDA or EMA can get a better understanding of the natural history of the SCN2A related disorders. And that can really help demonstrate unmet need. It can help design really meaningful clinical trials that address the issues that are important to families. And it can also give data to support biomarker development, which can help accelerate the drug development process. Really important. Next slide, please. And then, of course, a huge win going back to advocacy full circle is it can help foundations like Families SCN2A Foundation learn more about symptoms and related conditions and healthcare usage of the people that they represent. And that can lead to better outreach and better advocacy plans. And it can also lead to better collaborations with some partner organizations. If you start picking up comorbidities or seeing patterns with families with SCN2A related disorders compared to other groups that maybe you didn't know was a natural fit, because they weren't the same, near the same gene, or they weren't channelopathies, or they weren't otherwise associated, but you may start to see patterns with other disorders that may lead to some collaborations. For example, sleep studies, or other platform drugs, or lots of different ways that groups might collaborate. And this data could help give the advocacy groups some information that they need. So next slide, please. Okay, so cheers. It's lots of exciting, lots to celebrate, but the advocacy work is not completely done. We do get to take a pause, we get to pat and be excited, but in the next upcoming months, the foundation is going to take the lead in providing more education and promotion materials to families and also to clinicians to make sure that everybody knows the code and everybody's using the code correctly. Next slide, please. So what can families do? So the very first thing is remember your code. Again, we give lots of ways to remind you to use the code and help you remember, and information to give to your clinicians, but memorize the code or write it down and share it with every provider and every visit. And this is especially important with doctors that maybe, and healthcare providers that maybe aren't used to working with families with SEN2A related disorders. So, like if you're going to urgent care, if you're getting care out of town, if you're seeing a specialist that's outside of your normal circle, they really may not know the code. And so if you can help remind them to use it. So I made a little mnemonic device, because this is the way my brain works, to try to remember this code. It's 7 digits, so it's like a phone number, if you can remember the rhythm of a phone number. But in my head, I'm like, QA, questions and answers. I've got lots of questions and lots of answers. And then the middle one, the 0.0, if you could click on to make the next thing come. It reminds me of a smiley face. Whoever's got the, if you could click to the next part of the slide, it should add a little smiley face. There we go. and then another eye, and then the smiley. The 0.0 reminds me of a curious little smiley face. If you click it one more time, please. There we go. And then the 101, I think about like basic information. When you're learning something, it's like, learning SEN2A 101. See, it's like your primer. So to me, that's how I'm going to remember this code. QA, smiley face, 101. Next slide, please. Okay, for international families, so for those of you who are living outside of the United States, you can still share this code with your provider. They may jot it down. It may not be formally listed in your codes if your country hasn't adopted it, but it's still great to tell them. It's still great to be thinking about it, because at some point, hopefully, the next version of where you are will be using the new code. But for now, you can just share it. Just don't be surprised if you don't see it officially listed in the record. Okay, next slide, please. Okay, and save the date. The official rollout is October 1st, 2025. Again, there'll be lots more foundation push and education and clinician education before then, but if you could just mark the date officially, that's when you should start sharing it with your providers, if not giving them the heads up beforehand. Cheers. I think that is it. And perfect timing for me, because I have to get in line to get on a train, but I'm going to stay on the call just to forgive the movement on the on my camera, but I'll still be here. And I appreciate you all so much for letting me chat tonight.
Thanks, Melody.
Thank you, Melody.
Always a pleasure.
We needed to distribute celebratory kazoos to the audience for this one. At this point, we need to.
Yeah, go to React, and then you can do all kinds of fun things.
Yes.
I like that rocket one. That was fun. I haven't seen that.
I didn't put it together until now that that's Ben's birthday. I can't believe it. October 1st.
How
That
perfect is that, Leah? I love that.
So funny. He'll be 15.
Crazy.
Are there any questions about the ICD-10 or anything else that we've talked about tonight?
Thank you all for this amazing news. Hopefully we can keep getting our funding. We'll keep working hard to make sure we have funding.
Well,
Hey,
Victoria, you're doing your part to keep us to keep things moving and to keep advocating and awareness going. If you want to get an awareness day and recognized in your state, you can contact us and we'll put you in touch with Victoria.
Leah, I did have a quick question.
Of course.
And I don't know if this would be for Sean or for someone else. With the CRISPR stuff that's been going on, there is talk about that there is concern that it can cause things afterwards. So like you edit the SCN2A gene, but then it can have an effect that you don't anticipate happening. We know that the little kiddo was the first successful version of gene editing. Are there, sorry, Parker is not happy right now. Are there examples that are out there about just what could possibly happen in the other direction, just so we're prepared and aware of what that could look like? Things that could happen, they could become to cats, or who knows.
Really important question, Jessica. And Karen, maybe you and I can tag team this one. But I guess when we think about safety risks, I often think about things as, delivery risk and class risks, and then side effects. So for these modalities with the delivery, which will likely be a gene therapy for CNS, there are risks with that of a systemic inflammatory response that can be very dangerous. And we've gotten much better at being able to manage that delivery approach with the use of high-dose steroids and a few other medications. And also the warning signs where it's starting to become severe, where we may want to escalate that immune suppression protocols in different ways. But to your question more poignantly, with these gene editing, we're editing the genome. So there is risk of bystander. So that not only would it be changing and editing the one particular mutation that we're interested in, but there's some potential that it could mutate other areas of the genome. We are getting very good at identifying what other potential areas of the genome that it could or may potentially risk or edit. In the instance of baby KJ, there were a number of other sites that were potentially editable and were edited in most of the preclinical studies, but they were edited in such a way that they weren't dangerous. They edited one base pair for another that led to the same amino acid, so it didn't change the protein or function in a dangerous way. So maybe to answer that question, we're getting quite good at being able to pre-identify areas where you could have off-target effects in that regard, and I suspect we'll only get better as AI and time goes on. But there are other things to probably have considerations too. That Karen, what are your thoughts?
There's one other aspect that I did want to bring up, and that is that because of the way that CRISPR has to be delivered to the brain now, which the best case is the AAV or maybe some vector type based thing. Not only is there the inflammatory response, but there's also the hesitation to redose with these carriers, because these carriers could carry a risk of an inflammatory response the first time, but the second time you're actually re-exposing the body to the same potential allergen again. And then the second time, the immune system could really ramp up its response. So based on what I've read, that can be a potential risk as well. And so that means that if you are dosed with for example, an AAV9 vector carrying a CRISPR base editor, there might be the risk that a doctor would want to do that to the same individual again, even if the second generation CRISPR is perhaps more effective, or the capsid protein becomes more effective at delivering the cargo to more cells inside the brain. So there's that risk as well at this point in time.
Great, thank you.
I have a bit of a question. It might be a little convoluted and not entirely on topic with the codes that we were talking about earlier. I've been trying to understand for a couple of years kind of what's going on with my daughter. Her primary diagnosis is actually Aicardi syndrome. The SCN2A was sort of a secondary finding, and a lot of her doctors have sort of dismissed it as benign and just focused on the Aicardi syndrome. But we've seen a lot of med resistance that sometimes feels like it's a little more than what we would see just in SCN2A, certain reactions to different medications. And I only just found out a few months ago that apparently the genetic mutation came from me. But I still, like, I can't find enough information to sort of answer the questions that I'm not sure how to ask, if that makes sense.
Could I ask you whether or not the SCN2A mutation that was discovered, was it considered pathogenic, likely pathogenic, benign? Do you remember anything about that on the report?
Yeah, it said likely benign. It's the PARG28CYS. And I think there's a couple other letters in there somewhere too. R28C.
Sorry, PARG.
Here, let me type it in.
So we've got an arginine to something change.
And I don't know what my keyboard is. That's not supposed to do that.
Yeah, don't worry. My note taker just ran out of charge too. So we're all in the same boat. There we go.